Okay, hi everyone, welcome back. And if you've not, study guide is up. It's on Lima, and let's go there real quick. So if you go to resources under review guides, you can find it right here. So it'll be under review guides, and it says pre-Wednesday because we still have our Wednesday lecture. But again, after I'm done with today's lecture, hopefully I get through everything I have planned for today. So no top hat today because we kind of have to motor. We only have this lecture before exam 1. So this upcoming Friday will be a review session, and then next Monday, or on the Monday, the 14th, that's when we have our first exam. Okay, so again, I'll finalize everything that's in blue text at the end of this review guide here. And you're like, oh my god, this review guide is so long. Well, it pretty much is everything we did cover except for maybe the pineal gland and melatonin, but we did cover that during class. It's just that we covered a lot of ground. Okay, so again, don't worry about top hat for today because again, we're going to go real light speed. So I know it'll be kind of fast, but I'm going to try to get through this. Alright, so what is weird about this blood smear? I'm like, okay, I see a lot of red blood cells, I see some platelets, and I see some white blood cell here on the side. But what's weird is that some of these red blood cells have an unusual banana-like shape. And this is what we call sickle cells. So sickle cells, we have these normal red blood cells. Again, they have that round biconcave look right here. And yeah, I know. I was like, but the thing is that at least you'll be done with the exam, but then you get to enjoy whatever after. So you get to relax after the exam. Alright, so what we have here is what causes sickle cell is that a mutation happens in the gene for hemoglobin. So the other weird thing about this is that when you have this mutation, it changes the overall shape of hemoglobin. And now hemoglobin forms this like long, and it's inflexible, so it's more rigid. So just by changing the shape of hemoglobin, you change the overall shape of these red blood cells. And the thing is that, so again, you're just having one mutation, and that changes the shape of one gene, one protein, and that causes a change in the whole type of cell or shape. Again, this is why it's important to know the genetic code, and you don't have to, okay, for this class you don't need to know the codons, but you have to appreciate that with genes contain information, and then you have RNA, and any sort of alterations in your original DNA is going to cause a change in RNA, and therefore cause a change in protein. So what happens with sickle cell, and I don't expect you to know this particular mutation, maybe if you take a genetics class you have to know this, or med school you have to know this mutation, but what happens is that this is part of the sequence for hemoglobin, and in sickle cell disease, all that happens is that this one base pair right here, the sequence changes just by one, and that's able to alter the whole hemoglobin shape, thus altering the whole hemoglobin or the red blood cell shape, just by hemoglobin's gene having a variant that mutates it. Now, this is why it's important to appreciate the link between DNA, RNA, and protein. Now, sickle cells, what this means here, this like HbS allele frequency means like how often you find this in the human population over here. So what we have is Africa over here, and you see that this sickle cell trait is found a lot in African and sub-Saharan Africa over here, but also in the Arabian Peninsula and on the Indian subcontinent over here as well. But you also have some occurrence of this along the Mediterranean as well. But if you look at where, and this is one of the leading hypotheses, because what they saw is that, okay, that malaria is often common in these geographic areas. So the thing is that if you overlay these two, where malaria is frequent, and you also have this frequent variant of the hemoglobin that causes sickle cell disease, is that you see that they kind of correlate with each other. And it's not coincidental because people who have the sickle cell trait, they're more resistant to malaria. Now, what we have here is that the malaria parasite, do I want you to know this entire life cycle? No, that's for your microbiology class. But what you should know is that, yeah, for malaria is able to invade these red blood cells right here. So this is part of the life cycle of a malaria parasite right here. And so this is why it's very important this is why sickle cell disease is related to malaria. And what we have is like, okay, this is showing how this parasite, this is able to invade these red blood cells and replicate or use it as part of its life cycle. But with sickle cells, it has a harder time replicating, and plus, like, it targets it more for attack and degradation by your immune system and macrophages and phagocytes in your blood system, or in your circulating in your blood. So this makes this sickle cell makes malaria, or the parasite that causes malaria, more vulnerable to your immune system. So this is why people with the sickle cell trait, they have increased resistance. Now, again, this is, now, do you have to know Punnett squares? Again, this is like very basics right here. But what here we have here is like, okay, this blue indicates that, remember that we inherit 2 sets of chromosomes, one from mom, one from dad. And then what we have here is that, okay, these people carry the sickle cell trait, so they have one normal copy of hemoglobin, but the blue implies that they have another copy that's actually that elongated sickle cell hemoglobin. So just by pure probability and basic Mendelian genetics, that you have this high 3 out of 4 will have that at least one copy of this sickle cell trait right here. Now, again, this is a genetics class, but what's happening is that the people who are carriers of this variant of hemoglobin, they have increased resistance to malaria, and therefore the children that get at least one copy of this, they'll also have increased resistance to malaria. But the thing is that, why is it better to have a double dose? And this is what's related to sickle cell anemia. So again, normal red blood cells are round and biconcave, but sickle cells, notice that they're kind of elongated and they kind of shaped like, they say sickle like elongated and curved like a sickle. I like to say they're kind of like almost like banana shaped because they're elongated and kind of curved. But these elongated cells, like these little sickle cells, it's kind of like small little rafts. Like they can flex, they can bend, they can go through this well, it's not a lazy river in your circulatory system. But the thing is that round shapes have an easy time bouncing around and they're a little more flexible. But these it's like having a log jam or a bunch of branches in the stream, where if they're elongated, they can get trapped among each other and also trapped in these vessels like here. So the thing about that is, not only is it able to kind of like clog up a lot, but it's also more fragile. Like these round shapes right here, they're able to deal with more stresses and mechanical bouncing around your blood vessels and pressures better. But these sickle cells, it's kind of like a branch. Like it's easy to break a branch like this, right? Similar with sickle cells, they're more fragile than their round counterparts. So this is why that sickle cells also have an increased risk of hemolysis. Again, hemolysis meaning that term when these red blood cells burst and lyse. So this is what is going to cause anemia, just like how with iron deficiency anemia, you're not getting enough hemoglobin and you're not getting enough red blood cell oxygen transport. Well, if your red blood cells are bursting and having trouble staying intact, you're also going to develop anemia as a result. So people who have no normal blood cells and they just have mutant copies of this elongated hemoglobin, they're more prone to the sickle cell anemia. But again, so it's kind of a trade-off between like, okay, are you having increased malaria resistance, or are you going to have increased or also increased chances of anemia due to these variants of sickle cells being more fragile than normal blood cells. Okay, so we talked a lot about anemia, and this is again hematocrit. Hematocrit, this is why it's important to measure it, because again, hematocrit is mostly based on your red blood cell count. Therefore, it's going to affect your blood's ability to carry oxygen due to hemoglobin. So anemia is when we have this depressed hematocrit level, but what if you have too many red blood cells and it's like, okay, are you doping, or is there something else besides taking a lot of EPO that can cause a lot of red blood cells? Well, there's something called polycythemia. So poly, again, that useful medical scientific prefix meaning many. So this is what's happening here. It's the opposite of anemia. We have elevated, abnormally high hematocrit. Now, what are the effects and what can cause high hematocrit? Well, as a general overview, there are many different causes. I don't expect you to know all of them at this level, but things like dehydration, like say you have a soup and you leave it boiling on the stove for a while, what happens? You lose water from that soup, so the soup gets more concentrated. So you can have a high hematocrit if you're, or a slightly higher hematocrit if you're dehydrated. Why? Because again, things are getting more concentrated overall in your blood. Also high EPO, again, due to high elevations or doping or something that triggers high EPO production. That can also cause high hematocrit, like that cyclist that had a 60% hematocrit. Not sus at all. And there's something called polycythemia vera, and I italicize it because it's more saying, okay, this isn't due to just like random or just like fluctuations due to hydration or differences in EPO production, but it just means that this person is producing more red blood cells than normal. And what happens is that these blood stem cells in people who have this condition, they have these stem cells dividing and proliferating more than usual. Therefore, they produce more of these blood cells and formed elements, especially red blood cells, right? Because again, all blood cells come from hematopoietic stem cells. And okay, sorry, quiet, Mr. Peanut. I'm lecturing. Okay, so what else can cause it is low EPO as well, because it's kind of weird. I think like, why is it low EPO? It's kind of cut. It seems like it's contradictory, or actually, it's like it's not the cause of polycythemia vera, but just because like, okay, you have a lot of red blood cells already, so this is like a telltale sign of this polycythemia vera. It's like, okay, your kidney's like, all right, oh, I have enough red blood cells, I have enough oxygen, so I don't need to make as much EPO. So this polycythemia vera, low EPO is also another sign that goes hand in hand with having too many red blood cells. Yeah. And is it too good to have a high hematocrit? And the thing is that you might think, well, isn't it's great? Like now I have all these red blood cells and I have this super high hematocrit. I'm going to run a marathon and it's going to be super easy. Well, think of it this way. Like what happens if you have a soup and you add more chunky stuff to the soup? It gets thicker, right? So if you go past like the way upper limits of hematocrit and I should look up like what the actual limit is. But the thing is that if you, again, the thicker more solid things you have in a liquid and suspension, the thicker it gets, right? So your blood will literally become thicker, and also that the more cells you cram into one space, then it's also going to cause them to be more prone to hemolysis. So high hematocrit has its disadvantages as well. Alright, so back to a blood smear, and here we have a blood smear that's relatively normal, but how about this one? Everything, take a few seconds like, oh, what do we see way too much of here? And again, here's a hint. So here's a red blood cell, here's a white blood cell, and here's a platelet. Yeah, so what do we have what's wrong with this blood sample in the right of this image right here? There are too many of these cells that look more like white blood cells. Alright, yeah, Yep, so we definitely have too much white blood cells, and the right image was from somebody who has leukemia. So leukemia, there are 4 main types, and I don't expect you to know all 4 main types, but what happens, how is it categorized? Well, it depends on whether this abnormal amount of white blood cells was a sudden increase or if it occurred slowly over time. So what was something that happens in a short period of time, in general, that's a common term we use in medical terminology, meaning acute. So it's kind of like a sudden, short change. But chronic change is something that's kind of sustained over time. So was it a sudden change, or was it gradually increasing over time? And it also depends on characteristics of the white blood cells. That's why these are abnormal white blood cells. They don't really fall neatly into those 5 categories of white blood cells. And there's myelogenous and lymphocytic or lymphoblastic. So again, you have so this is how you have the 4 types. You can have acute myelogenous leukemia, you can have chronic myelogenous leukemia, you can have acute lymphoblastic leukemia, you can have chronic lymphoblastic leukemia. So this is like 2 by 2, you get 4 main types. Now, myelogenous and lymphoblastic, that kind of looks like the myeloid and lymphoid. And this is what determines those two, like those categories that have myelogenous or lymphoblastic or respectively, because it determines based on this. But again, this is why I italicized it. I don't expect you to know it at this level, because that's pretty, like if you're going to like oncology or blood cancer research, then that's more pertinent to your interest. But for now, just know that this is where we get that subtypes of leukemia. Yeah, so if you have high white blood cell count, does that mean like and this is why they do cell blood counts when they do a blood draw, and you might see that they count the amount of red blood cells, white blood cells, platelets, and you might be like, oh, my blood count ended up saying I have high white blood cell count. Does that mean I have leukemia? Should I consult an oncologist right away? Probably not. So the thing is that most likely hit the most common cause of elevated white blood cell count are infections. So that just means that your immune system recognizes that you have some sort of pathogen circulating in your body, so it's going to ramp up the production of all those immune cells so it can deal with it. So that's a good thing. And this is why it's not always like, okay, you have the symptom or you have this test, therefore you have this disease. It's about probabilities. So again, even though both of them have elevated white blood cell count in common, which is more probable? And this is why you also do additional tests as well. Yep, so yeah, why high white blood cell count can also be caused by autoimmune diseases as well. So this is why it's like a lot medicine is not simple diagnosis, like we just take one piece of evidence, therefore you have this disease. You often have to take multiple things into account. All right, so, and leukocytosis, this is the term for a high white blood cell count. And leukemia refers to the cancer involving these white blood cell like tissues or cells. So high abnormal white blood cells. And again, is it more myelogenous? Is it more lymphoblastic? That's why they have those subtypes as well. And sometimes this is also why leukemia is sometimes called blood cancer. I think that's used more in like UK and other places. But yeah, so leukemia is not the same as leukocytosis. So when you have infection, you have leukocytosis, not leukemia. And leukopenia is a low white blood cell count. So again, it all has the root word leuk in it, meaning white. But the thing is that, yeah, that suffix, the ending part, they sound the same, but they're not the same. So again, just like thrombocytosis and thrombopenia is abnormally high or an abnormally low platelet count, respectively. And yeah, so high white blood cell count, it might be a cause for alarm because yeah, it might indicate, it's commonly an indicator of infection. But if you look for infection and it's not there, it's like, oh, maybe something happened where they just have a high, let's monitor it. So that's why they might do a follow-up blood test or see if that was a problem with the machine or something else that could. So again, this is why sometimes when they try to find something and they don't find what they think it is, you might have a repeated test as well. And and if you hear random burp noises, sorry, that's Mr. Peanut. He's molting and he's a little cranky today. All right, so hemophilia. So hemophilia is not just one disease, it's actually a group of diseases, just like leukemia is not just one disease. It refers to a group of different types of disorders involving bleeding. And what can happen with people who have hemophilia is that they can get something like a paper cut, and for most of us, we can just take a Band-Aid or just even take a tissue, apply pressure, and we have hemostasis. But these people, even a small cut can result in them just bleeding out, and they're unable to form clots like the rest of us can. So yeah, this is like what it might look like if someone has hemophilia and just a small cut, but because they have an inability to form clots properly, they can end up bleeding out. So there are different types, A, B, and C. Do I want you to know the difference between the three types? No, this is not a med school class. And the interesting thing though is that men are more at risk for A and B. And why is that? Well, the thing is that many of the genes that are involved in these clotting factors, there are some that are linked to the sex chromosomes. So there's X chromosomes and Y chromosomes. But the A and B, genes that cause A and B, these are on the X chromosome. So men only have one X chromosome, women have two. So you can have an abnormal one, a lack of this clotting factor. If it's a woman with like a lack of it on one of her X chromosomes, at least she has the other one. But a male with the XY, if they have a lack of this clotting factor, then they can't get it from their Y. They're stuck without a clotting factor. And this is why it's important to know, this is why the coagulation cascade is super important for medicine, because again, if you have problems with the clotting cascade, you can't form a clot. And this is the difference between the three types. Again, I don't expect you to know this, but I want you to appreciate that if something happens along this way, where especially for things like factor 8, which is important for forming factor 10 and activating it, if you have an ability to form these factors and activate these factors and enzymes in the coagulation cascade, you're not going to get that fibrin clot. So this is what happens in hemophilia. These people have impaired clotting because they have problems along this coagulation cascade. Now, how do you treat that? Well, the common treatment for hemophilia, so people who have hemophilia, they typically carry around a little medical kit containing recombinant clotting factors. So they have pretty much the clotting factor they're missing in a little vial that they can inject in case they end up having a cut. So how do they do this? They take the human gene for the clotting factor they're missing, and then they put it into bacteria, and the bacteria are able to crank out this protein. So they just, again, DNA makes mRNA, makes protein. So these are why genes are very important, and knowing that link between DNA and protein is important. So you put it into this bacteria, and these bacteria are just meant to crank out proteins. So what we have here are these little plasmids, and they just take the gene, put in this little loop of DNA called plasmid, and the bacteria just try to just crank out the proteins from this plasmid as much as possible. But you don't inject the pure bacteria, because again, bacteria are foreign to our body, so that's going to cause a crazy immune reaction. So what you do is like grow this bacteria, extract the proteins, purify the proteins so it doesn't cause immune problems, and then this is what people who have hemophilia carry around. It's just like insulin. People who are type 1 diabetic, they carry around insulin to help them because they lack the insulin. Same with hemophilia, very similar strategy, just with a different gene. Okay, so now let's talk about blood types. Ooh, yeah. So I already saw some blood type questions and you had this in the lab as well. But this is important because, again, and especially with everything COVID related, we've had a lack of blood donations because it was hard to access, and just safety regulations as well. So we have a lack of blood in general. It's like not just in Hawaii, but just like pretty much nationwide, I'm pretty sure worldwide as well. But what are the main blood types? So we're just going to cover the very basics, the ABO-RH system. And what we see is that we have our red blood cells, but we have something on them called surface antigens. So these are little markers or little ID tags that are unique based on your... Now, ABO isn't the only type of blood type and antigens, but this is like kind of like when they were first discovering that they couldn't just transfer blood from anybody into anybody. This is one of the first discoveries they made during the system. So it's not perfect, but does help screen things. Now, this is what happens with ABO blood groups. If you look at the red blood cell types, and people with type A red blood, they're going to have the A antigen on their surface. Come on, pointer, come through. Here we go. Okay, so they have this A antigen on the surface. People with type B blood, they have the B antigen. And then people with type AB, they have both the A and B antigen, whereas people with O, they don't have the A or B. But notice that it's not completely bare and without any antigens. There is an O antigen. But in terms of the simple ABO system, what happens is that you produce antibodies. If your immune system is behaving normally, you produce antibodies against things that are foreign to your body, but you shouldn't produce antibodies against things that are naturally found in your own body. So this is why people with type A blood, they're producing anti-B antibodies because they don't recognize this in their body. But they're not going to produce anti-A antibodies because then they would attack their own body and tissues. That's an autoimmune reaction. If you attack, your immune system is creating antibodies or creating cells that attack your own tissues. So it's very important that your immune system recognizes things that are foreign, but doesn't attack itself and attack your own cells. Now, what are these antibodies? What are these immunoglobulins? Are they different? Are they actually the same? So again, antibodies is just another name for immunoglobulins. So if you hear me, I use them interchangeably. So if you hear me talking about one, I'm talking about the same thing. So I use those two as synonyms. But These are used by your immune system. Again, we're not going to talk about everything about antibodies right now, but it's important to understand antibodies to understand blood typing. So antigen is not the same as antibodies. So immunoglobulins are the same as antibodies, but antigens are different. Antigens are what antibodies recognize. So here we have antigen, and an antigen is some sort of molecular surface, and the antibody is recognizing it. So it's kind of like a lock and key. It's kind of like a hormone and its receptor. You have an antigen binding to this protein we call an antibody. So antigen is what an antibody recognizes. Or you can think of it as that antigens generate antibodies against that antigen. So yeah, that's why it's like the antigens are there, like what causes antibodies to be made. Now antibodies, they are represented as cartoons in many forms. Like this is like probably the simplest version, like a little Y. And why is that? Well, you have something called antigen binding surfaces. So these are the parts that can attach to antigens. And again, this is like these antibodies, these immunoglobulins, they're proteins. So technically, in theory, if it has a chemical structure, you can have an antibody against it. So what happens is that these binding surfaces kind of fit depending on the shape of the the shape of the molecules of the antigen. These kind of antibodies are supposed to kind of fit them like a glove if they're able to target these antigens. And this is a more detailed version. So this is showing more of like the ball model or space filling model where there's, again, antibodies are immunoglobulins and they are proteins. So this is showing more of the structure, but what we have here are the binding surfaces. So per antibody, like simple IgG we're seeing here, we're seeing this, that there are two binding surfaces in a simple antibody like this. Now, with the antibodies, is that they recognize things called, the surface that they bind to on an antigen is called an epitope. And I know it's a lot of new words, but there is a difference between epitope and, so again, antibodies recognize antigens, but where they meet is what we call an epitope on the antigen. So how do they bind it? Well, they bind via these chemical bonds. So it can be ionic chemical bonds. It can be also what we call van der Waals forces. And basically it kind of fits kind of like a little, just like hormones bind to the receptors, it fits into a little pocket that's meant to kind of cradle the antigen. So again, this is looking at the antibody and the two surfaces it binds to. And we're seeing an antigen kind of fitting into that binding site of an antibody. And if we look even closer, or actually this is the OpenStax version. So OpenStax is showing you yet another version of the protein structure. So this is the space filling where each of these balls is a little atom. But this is kind of showing you like, oh yeah, there's the secondary structure, protein structures to the antibody. And you have these binding surfaces here. So it's kind of, this is why it's often represented as that like fancy Y-looking sort of structure. So yeah, this is the antigen binding site, and what you notice is that it's not, even though it's a protein, this is why it's symmetrical, because you have two heavy chains and two light chains, and with one heavy chain and one light chain, so the antigen binding site isn't just only on one of these chains, it actually shares like the whole part that an antigen binds to is made out of one of these halves of heavy chain and light chain. So it's actually made out of like 4 separate polypeptides. Now, what we have here is a close-up of this antigen, something that's foreign to your body and being recognized by an antibody. So it's a foreign antigen being recognized by an antibody. Now, this surface right here is what you call an epitope. So this is a part of an antigen that antibody recognizes. So it is possible to have multiple epitopes per antigen. So this epitope is this surface here for this antibody, but it is possible to generate another antibody that recognizes this surface. You could have an epitope here, you could have an epitope here, you can have an epitope here. It's kind of like grabbing onto grabbing onto any object here. So if you have an object, you can grab it onto it like this surface, but this can be epitope, this can be epitope, this can be epitope. Same antigen, same object, but I'm grabbing onto different surfaces. So epitopes are different surfaces. I know. So I'm not flexing here. I'm just this was something grabbing onto with so that's what we have an antibody. So again, Antigen is something foreign, but epitope is what the surface that is being grabbed onto by an antibody. So I also like to think of antibodies as like little people, and again we have two hands, so we're able to grab onto something with each hand. Same with the antibody. Antibody is able to grab onto something with each of its binding surfaces. Alright, so again, immunoglobulins. And antigen is something that can cause an immune response and create antibodies. Again, if it has a chemical structure, in theory, it can generate your immune system can generate an antibody against it. But epitope is a specific part of an antigen that is being grabbed onto by this antibody over here. Alright, so again, this is a very simple version. So this is more like a PHYL 142 version of the antibody. Again, I don't expect you to know all the heavy and light chain. If you take biochemistry or advanced cell biology, you definitely will have to know that stuff for sure. But again, antigen binding sites 2 per immunoglobulin. And so if you have something like a viral protein I'm showing you here, then per antibody, you can bind up to these two proteins on their epitopes over here. Now, with these proteins and this, so here we have all these like viral proteins and particles, and you can have antibodies that join them. But the thing is that you can also have multiple antibodies recognizing that same protein, and this is what we have in this example. So you can have what we call cross-linking, and you can have multiple antibodies forming these vast clumps and networks of antigens binding to antibodies, and all this like kind of daisy chains and branch networks of this antigens and antibodies binding. And this is very important because again, antigens, antibodies, this is how your immune system is able to kind of like, or let's rewind a bit, I'm kind of getting ahead of myself. But what we have here is like now this viral particle is being surrounded by antibodies. So this is one way your body is able to neutralize the infectious particles, is by coating them with antibodies. The other cool thing is that cells of your immune system can also grab onto antibodies and help neutralize and dispose of these particles and foreign pathogens even more, because the immune system makes extensive use of these antibodies. So it's not just about coating foreign things, it's also about helping your immune system along. Now, here's an infamous virus, and what were we going to do? Well, are we going to generate the same type of antibodies that we saw in the previous example? Well, yeah, we can have an antibody that was generated in the previous example, but this is a totally different shape. So this epitope over here with this viral particle, that's not going to be recognized by this antibodies. And again, disclaimer, these antibodies aren't to scale. Like antibodies are really tiny compared to like cells and viral particles. I'm just showing you so that you're able to see the surface even better. But back to this, like we have that antibody recognizing this virus, but this virus and this viral proteins are a totally different shape. So this antibody isn't going to recognize it, and antibodies have something called specificities. So one antibody isn't going to recognize all different types of foreign pathogens in your body or that happen to enter your body. Antibodies bind to the surfaces that they mold and fit and are able to bind to via these chemical bonds. So they are specific to specific antigens and epitopes. Yeah, so the thing is that they could have, what we have is like, so we don't have this antibodies binding, so different virus, we have to have a different antibody, right? So we need a different antibody that recognizes the surface of this new virus, and that's what we have here. So notice that the shape and the binding site of this new antibody isn't the same as in the previous example, because again, the previous example doesn't really fit onto these viral proteins. So you need an antibody that has a shape and binding site that molds better to that viral proteins and surface. But topical, what if this virus starts to mutate? And remember that nucleic acids, like, not to get too much into detail, but viruses can actually have DNA or RNA as their genome, but either way they're going to make proteins from nucleic acids. If these nucleic acids such as DNA and RNA start to change their sequence and mutate, that's going to change the proteins on these viruses. Ooh, I had a really cool thing. Let me fix something real quick. Right, because it's a new slide I have. Oh, that's it. Okay, let's restore it. Okay. Alright, so what if this virus starts to mutate? And what happens if it starts to change these nucleic acid sequences? Well, that's also going to change their surface, could potentially change their surface proteins as well. So if it changes their surface and it changes their appearance, it's kind of like the virus gets a glow up. And it's kind of like, okay, but now what do we have here with these new surface proteins? Notice that the old antibodies aren't binding to it as fitting onto it as well as the previous version of the virus. So these antibodies aren't going to be as effective because they can't grab on to these new surface proteins on this virus. And this is why these COVID protocols and these new variants, even though we're vaccinated and hopefully you're boosted, but this is why our original vaccine was made for the original version of the virus, but since the virus picked up additional mutations, the surface looks a little different, and that's why our old antibodies to the original virus might not recognize them as well. Yeah, so it's like classification of the virus. Yeah, that's what it is. Miss Rona got a glow up. It's so annoying, but then she's gonna do what she wants, and she's like, no, to your new antibody or your old antibodies. Is there some sort of mutation where there's different antibodies on the same structure? I don't know about, like, if you talk about whether antibodies can bind to the same structure, you can have antibodies, like you can have antibodies, like say you have new antibodies that bind to this new part of the spike protein, or if you bind to this new spike protein. So you can have different types of antibodies binding to the same original structure. And actually it's really cool, like this is how your body is able to generate so many antibodies. It kind of has like this like arsenal, there's like this deck of all these protein structures, and it's able to kind of shuffle it around. It's kind of like a random number generator, and then by generating all these different shuffling these parts of the genes around, it's actually able to generate, well, not necessarily infinite, but a large amount of combinations that might as well be infinite that can bind to different types of surfaces. Yeah. Okay, so back to blood type. So blood type, ABO, blood group, is that what we have, and this is the OpenStax version I'm showing here, but what we have here is like the Martini version. So surface antigen A, this is what makes someone a type A blood type. So they have this something called antigen A. Now type B is an antigen B, surface antigen B, which has a different structure than type A. It's actually very similar, but for simplicity's sake, they're going to make it very different. Now, are all antigens different, or actually I may have like contradicted myself earlier, but antigens aren't necessarily foreign to your body. So these surface antigens, these are natural to your body. So you have antigens that are natural to your body, they're endogenous antigens, but you also have foreign antigens. So your body gives your immunoglobulins in your body, they shouldn't recognize your own antigens, but they should attack they're generated against antigens that don't belong or aren't naturally found in your body. So people with type AB, they have a mixture of these A and B antigens. And then people with type O, and this is where the OpenStax is a little better than Martini, because type O, the Martini version implies that there are no antigens, but that's not quite true. It just means type O just means a lack of A or B antigens. And yeah, this is why I think the OpenStax is a little better, because it's just saying like, yes, it just has a lack of A and B. It doesn't mean that this red blood cell has a totally naked surface. Now, these antibodies, notice that you're generating antibodies against antigens you don't have on your own cells. So this is why people with type A, they generate anti-B because they don't have B antigens in their body. People with type B blood cells, they generate type A because these A's aren't found on their cells. But people with type AB, they don't generate anti-A antibodies. They don't generate anti-B antibodies because then these antibodies would attack their own cells. But people with type O, and in terms of ABO blood type, they generate both anti-A and anti-B because they don't have either. They don't have the A antigen, they don't have the B antigen. So this stuff is foreign to their body. So it all depends on which antigens are naturally found on your red blood cells. Okay, so then we're a type A red blood cell, and things that so you might be like, wait, I have never had a blood transfusion. Why am I generating antibodies against B type, or how do I generate antibodies against B antigens when I've never had a blood transfusion? Well, the thing is that there are things in the environment, including some bacterial chemicals and byproducts that kind of look like human red ABO antigens as well. So we encounter things that are A antigen and B antigen like during our daily lives, but the thing is that we're not going to generate antibodies against surface antigen A in this case. Why? Because then we would be attacking our own cells. So your body produces antibodies against foreign antigens. These are your natural antigens on your red blood cells, but if you have things in the environment that look like a B antigen, your body is going to generate anti-B antibodies if you don't have type B blood. Type B or type AB. Now, same with type B. So again, just like B antigen or B-like antigens are found in our natural environment, same with A-like antigens as well. So we have these A-like antigens, and what is this person with type B blood going to do? Well, they're going to generate antibodies against this A-like antigen. This is why people with type B blood, they have anti-A antibodies. Again, these anti-A antibodies are specific to the A antigen, therefore it's not going to attack their own cells as well. And then people with type O, again, misconception with the Martini version is that it looks like it's naked, but it just means a lack of A and a lack of B antigens. So what happens is that, again, we have A and B-like antigens in our natural environment. So people with type O blood, they're going to generate antibodies against the A-like antigens, and they're going to generate antibodies against the B-like antigens. So this is why people with type O, they have anti-A and anti-B antibodies. Now, this is where it's like, then this is why I think the OpenStax version is a little better than the Martini version, because this is what the A antigen looks like. It's actually, you might be like, oh, hexagons, kind of like glucose, right? Yeah, so it is some sort of chain of carbohydrates. We have a branch chain of carbohydrates. Do I want you to know, I even put this in the review guide, is that I don't want you to know things like N-acetylglucosamine or fucose or galactose we cover in nutritional context. But in terms of knowing the exact chemical structure, this is, I think, more appropriate for biochemistry. So don't worry about it for this class. But what we see with this B antigen is just swapping out a different type of carbohydrate is being attached to this structure, and this is why these antigens are on the surface of your red blood cells. This is a phospholipid membrane, and what we notice is that these antigens are actually embedded in the plasma membrane of our cells. And then we have this type O antigen, and again, this is why type O blood does not mean you have no antigens whatsoever. Type O actually has a very similar structure, it's just that it's lacking this little extra sugar that the A or B. So yeah, that's the only difference between A and B antigens, it's just a different sugar, and type O is lacking that additional sugar on this carbohydrate chain. So again, type A, this is what I'm trying to really drive home. Type O, it just means a lack of A and a lack of B antigens. It does not mean that your red blood cells have no antigens at all. So type O red blood cells do have this antigen, it's just not A and it's just not B. But the thing is that these type O antigens, they cannot be recognized by anti-A and anti-B antibodies, so it kind of gets a free pass on that. Now, there are other rarer blood types, but I want you to know the ABO-RH system, not the rarer blood types that are found in only a few people. So the ABO is the most common and was one of the first discovered type of blood types there, and different, like, and this human blood typing system is different from like Mr. Peanut's going to have a different blood type than I'm going to have. And I think someone asked a question about dog blood types a long time ago, and it's turned out to be a pretty big rabbit hole. Like, dog blood types apparently are very complex. So these right here, I'm just wanting to show you that there are more blood types like out there, but we're only sticking with ABO for this class. Now, RH is also a sticking point where people get confused, and Martini doesn't really have a picture of this, and RH refers to something called this, it's a different type of surface antigen. It's not, it's actually a protein. So this is what we call the RH factor, or sometimes called RHD, because it refers to this antigen D. Now this is completely independent from ABO, so this is not the same thing, it's something totally on its own. Now, RH right here is that this is, again, it's going to be a surface protein. It's going to, or not, or it is a surface protein, but it's a surface antigen. happens to be protein. So Rh negative, when somebody says they're A positive, or when they say they're A negative, the positive and negative part refers to the presence of these antigens. So if you're negative, that means you have a lack of these antigens, like we see on these cells here, the cell here. But Rh does not mean, negative does not mean no antigens at all. It just refers to the lack of this specific Rh factor. Now, this is where I have to people get a common sticking point, and I often see a lot of confusion. So ABO is different from Rh. It's not the same thing. Just because you have type A blood does not mean you're Rh positive. Nor does that mean, like, if you have type O blood, you're O negative. Things that blood cells that have A antigens on them does not mean you're A positive. You're A something, but are you positive, A positive, or are you A negative? It depends on that Rh. So positive and negative in this ABO-Rh system refers to this factor only. It doesn't matter what the letters are. It just depends on whether that Rh factor is on the surface of your red blood cells or not. So if there's Rh factor on your red blood cells, you're positive. If there's Rh on your no Rh on your red blood cells, that means you're negative. So if someone has A antigens but they don't have Rh, that means they are A negative. Alright, so to put it all together, so again, A antigen just means you have red blood, or type A blood means you have A antigen on your surface. And then with A positive, that means you have the Rh factor. So that's the difference between people who have type A positive blood and type A negative blood, is the presence of this Rh factor, whether it's there or not. Same with B antigen. The difference between B negative and B positive is that if you have the Rh factor, you're B positive. If you have B antigens but no A antigens and no Rh, you're B negative. Same with AB. If you're AB negative, that means you have A antigens and B antigens but no Rh. If you're AB positive, you're 3 for 3. That means you have all 3 of the antigens we talked about. O negative, again, doesn't mean a lack of antigens altogether. But in terms of A antigens, B antigens, and Rh factor, you don't have any of those. But if you're O positive, you don't have A, you don't have B, but you still have that Rh factor. So that's what O positive is. So this is the system I want you to know for this class. So ABO, and again, R and Rh. So again, with type A and type B, so remember that, what about people with type AB blood? Do they produce antibodies against anti-A or anti-B? They don't. So if they did produce anti-A and anti-B antibodies, well, say this person with type AB whatever blood, we're not talking about Rh factor in this example for simplicity's sake, but say someone with type AB blood, they somehow, their immune system started generating anti-A and anti-B antibodies. Well, it's going to recognize these foreign antigens, but it's going to also attack its own cells, causing it to lyse and rupture. So this is what we have with this is why it's important for blood transfusions to kind of assess whether you could have this type of reaction as well. Yep. So what we have here is agglutination. So this is what we have is like a surface antigen, and we have these antibodies. Say this person has a transfuse, or say this is like a patient with type A blood, and then they're getting, why is my mouse like so twitchy today? All right, so say this is a patient with recipient with type A blood, but they have anti-B antibodies, and they're getting transfusion of red blood cells with the B antigens on them. Well, that's going to cause agglutination, this clumping, and the immune system is going to eventually cause these red blood cells to lyse and rupture. So this is what we call, so yeah, I like to think of it this way. So this is like a little inner tube right here. And what we have with agglutination, like if you have like a little flotilla of inner tubes, is that all these people are linking hands and grabbing onto each other. So I like to think of agglutination kind of like that, a little analogy. So what we have here, all these red blood cells being held on together and clumping together with all these antibodies linking them. Sort of similar with what we see here. Agglutination is this clumping of red blood cells due to antibodies cross-linking and holding them together. Now, common sticking point and point of confusion is agglutination and coagulation. You may be like, oh, it sounds similar, but in terms of hematology, agglutination is referring to this clumping due to these antibodies and cells. coagulation is reserved for that coagulation cascade and that fibrin clot formation. So even though we do have aggregation or this gathering of cells, this agglutination is referring to this antibody reaction. Coagulation involves those clotting factors. So think of it, coagulation, clotting. Agglutination is antibodies. So antibodies, agglutination, coagulation, clotting. So they're not the same thing. So, and sometimes agglutination is also called clumping in our textbooks as well. Alright, so again, what happens if you donate type B red blood cells to type A patient? Remember that the type A patient is going to generate are they going to generate anti-A antibodies? No, again, that would attack their own cells. So this is why you don't have anti-A antibodies in a type A patient's body normally. Now, with the transfusion, what's going to happen? Well, we have things that people with a person with type A blood, they're naturally going to produce anti-B antibodies. So if you introduce anti-type B red blood cells and donate them to a type A patient, what's going to happen is that these donated red blood cells are going to be attacked by the type A patient's antibodies. So what's that going to do? It's going to cause agglutination, and it's going to cause clumping, and it's going to cause hemolysis. Yeah. Hello. Yes, trying to drop in on the lecture. And this is what we call a hemolytic transfusion reaction. So giving red blood cells, this is why it's important to know blood type, because you don't want to have this happen and cause the red blood cells you just donated to a patient to all of a sudden burst, because you want the red blood cells to stay intact. And again, hemolysis, important word, is a rupture and destruction of red blood cells. So many different ways to destroy red blood cells, including giving the wrong blood type to the wrong people. Now, symptoms, the thing is that your red blood cells, basically your body, when your red blood cells and the contents of your red blood cells are being spilled into your bloodstream, your body just goes crazy. And this is what we call a hemolytic transfusion reaction. So it's going to cause your nerves and your nociceptors to feel pain. You're going to end up with all this heme and blood in the pigment in your urine. You're going to get chills, and you're going to feel faint and dizzy because, again, you have all these red blood cells popping in your body and not enough oxygen being transported, and on the neurological effects as well. So yeah, it causes this crazy immune reaction. Your blood is this is what happens this is why it says febrile. Flank pain, pain. I've luckily never had this, but they say this is excruciating when this happens, this hemolytic transfusion reaction. So yeah, you're going to get all the drop in blood pressure, increased breathing rate. You just get systemic chaos in this transfusion reaction. And jaundice as well. Remember that jaundice is caused by the breakdown of heme pigments into biliverdin, then bilirubin, and this bilirubin accumulates in your body and goes to places that it doesn't normally go. So this is why it's important to match blood types. And is it typical to, and let's see, so this is why, so most of the time they try to match you I'm getting ahead of myself. So you separate red blood cells before transfusion. Why? Because again, you don't want the anti-A or anti-B antibodies coming along for the ride. So you kind of separate them using, how do you separate them? You can easily do this, your little centrifuge. And then what you can do is things that, because a person with O red blood cells, they have a lack of A and a lack of B antigens, you can easily transfuse these red blood cells or you can transfuse these to other blood types. Because, again, there's no people with type A blood and type B and AB, they're not going to have anti-O antibodies. And because these don't have A or B antigens, this is why their antibodies, the anti-B antibodies and the anti-A antibodies here, they're not going to attack these O red blood cells. And A can also donate to AB, and B can also donate to AB, because why? Well, with AB, they're not going to generate antibodies against A, and they're not going to generate antibodies against B antigens. That's why they can also receive from A and B as well. Now, Rh and Rh positive, it does depend on whether you've had a transfusion before or not. So, but in general, if you don't know whether someone's had a transfusion or not before, people with Rh negative blood, they're going to generate antibodies against Rh positive, or the Rh factor. But people with the Rh factor on their red blood cells, they're not going to have anti-Rh antibodies. So this is why you can go from Rh negative to Rh positive. But you can't do, and this is why I put the little asterisk or the little dash line, because you can actually donate for the first time from Rh positive to Rh negative. But after that first time, this Rh negative person is going to generate anti-Rh antibodies. That's why subsequent and the second transfusion reaction becomes riskier, because they're starting to generate immune response against this Rh factor. And this is why this person, you might have heard the term universal donor and universal recipient. So this is why O is considered the universal red blood cell donor, because again, they don't have the A or B antigens, therefore they can't be attacked by A and B antibodies. And AB is considered the universal recipient for ABO, because why? They don't generate anti-A antibodies because they have that, and they have A antigens on their cells, but they also don't generate B antibodies because they also have the B antigen on their own cells. So this is why they can receive from all of these four. And pregnancies, yeah, so that's where it gets tricky too. Like say a pregnancy, the Rh also involves the erythroblastosis fetalis, because again, if a mother is Rh negative, but due to genetic contributions from the father, that the baby developing in the womb is Rh positive, the mother's immune system is going to recognize that Rh factor as something foreign. So this is why the first pregnancy with an Rh positive baby in an Rh negative mother, that doesn't cause them to attack, or unless their immune system is able to generate enough antibodies in time. But that's why every consecutive pregnancy with a Rh positive baby in a Rh negative mother gets more risky, because she's starting to develop antibodies that can cross the placenta and attack the developing baby in the womb. Yeah. Yep, so people that are pregnant with children that does not have the same Rh factors. Yeah, so this is why it's also important, especially to genetic counseling is important, because you don't want loss of future pregnancies accidentally because like there's incompatibility this way. But if they have the same type of Rh factor profile, then they're fine, or at least in terms of Rh factor. But yeah, that's why it's important to, if someone knows their blood type and they know they're Rh negative, and they know their partner's blood type and they're Rh positive, then this is why they might want to see a genetic counselor to make sure that they have they come to terms successfully and don't harm the developing fetus and baby. Yeah, so yeah, but that's a little like I mean, it's good to know though, but yeah, it's also why it kind of amazes me how many people don't know their blood type, because again, especially in this case, especially if you're having to have children in the future and you're Rh negative, that can affect you as well.